Hello guys, I am Shubham from Food Tutorials. This video is about homologous recombination. So before we start, subscribe to our channel and press the bell icon to get notified whenever the video is out. So let's get started with the video. In the previous video, we have discussed about a double strand break repair pathway. In that video, I had told that the double strand break is repaired by the two ways, homologous recombination as well as non-homologous end joining. So in the previous video, we have discussed all about non-homologous end joining pathway. Now this video is about homologous recombination. So what is this? Basically, this is also a repair pathway, major repair pathway for double strand break. Okay. TSB repair pathway. Okay, so this is the first benefit of this homologous recombination. And the second is homologous recombination is also done in meiosis, right? That is known as meiotic recombination. In that, what happens? Homologous chromosomes pair to each other, right? Pair to each other, and it gives rise to new combinations. Okay, crossing over take place. Crossing over and exchange of genetic material. Exchange of DNA, right? So this takes place in the meiotic recombination. In this meiotic recombination pathway, double strand breaks are introduced with the help of proteins, right? So there are proteins which produce the double strand break artificially. Double strand break is not present in the DNA, but during recombination step, the breaks are introduced. From this, we understood that double strand breaks are also repaired by this process of homologous recombination, right? Homologous recombination is done by the holiday model. Holiday model. What is this holiday model? Because there is formation of holiday junction. Holiday junction. So what is this holiday junction? We will see. The first step, as we saw just now, homologous chromosomes are aligned in the first step. Second step, what happens? Introduction of double strand break. Introduction of double strand break. So double strand break is introduced with the help of proteins, right? Basically enzymes. Then is strand invasion. Strand invasion. Next is formation of holiday junction. We will see what is it exactly with the help of diagram. And the last step is the resolution of the holiday junction. Resolution of holiday junction. Got it? So let's see what is this. First step, homologous chromosomes are aligned. Homologous chromosomes are those which are similar. So these are the homologous chromosomes. This will come near to each other, right? Then the crossing over, exchange of chromosome will take place. Then the introduction of double strand break. Double strand break will be introduced. Then the strand invasion will take place. Okay, let's see with the help of diagram what is this. So we can see here double strand break. Okay, after double strand break, processing takes place. Okay, processing to generate single strand tails. Okay, overhangs basically. So these are the overhangs. These are generated. Then strand invasion will take place. Strand invasion of 3' end. Okay, strand invasion means what? This overhang will move inside the other homologous chromosome. Alright. So this is strand invasion, means invasion getting inside, invaded into the other homologous chromosome. Right. Then as the strand gets invaded further, the other homologous chromosome will also come upwards. Right. Then the strand will be synthesized. Okay, DNA synthesis will take place. So this is how double strand break is repaired. Then the next step is the branch migration. Branch migration is what? The strand enters, then this will get migrated. Migrated means moving further. Okay, it will move in the further direction. Like this. Okay, then after branch migration, this is known as holiday junction. This structure like X. This structure like X. This is known as holiday junction. Okay, so this is a newly synthesized DNA. I will write here. These structures, X-like structures, are known as holiday junction. Holiday junction. X-like structure crosses. What crossing over? That structure is known as holiday junction. And this recombination is accurate process. Okay, means in the previous video about non-homologous end joining, we saw that it is a mutagenic process. Means mutations are still there in the DNA. Mutations are not repaired. But in this situation, in the recombination, homologous recombination, the DNA synthesis is very accurate. Okay, so these are the steps, but basically every step requires some of the proteins, right? Proteins or enzymes. So let's see how this is whole process in detail. So basically this slide is on the resolution of the holiday junction. So how holiday junction is resolved? Basically, there are two methods. See, when we change the orientation, this has to be imagined. Okay, this has to be imagined, and how it will be visualized. Here is the holiday junction present, right? Now what happens? See, this is the structure accurately. So in the first step, the cut takes place like this. In this, we can see here. And the second step, it is vertically. Okay, step 1 horizontally, step 2 vertically. And then this gives rise to two products. After horizontal cleavage, the products are known as splice or crossover product. Splice or crossover product, reassortment of flanking genes. And the second step gives rise to patch or non-crossover products, no reassortment. Right? So in the first step, now how to remember this? So this is very easy. We have to apply the trick. So what we saw, two products are seen. After horizontal cleavage, we see splice or crossover products. And in vertical, we see patch or non-crossover. So how to remember this? We should remember by the word patch. Okay, patch. This word patch. Now, what happens in patch? Carefully note. In patch, see, The strands are like patch here. There is patch of what we can say other DNA. Like this we can remember. There is patch present of other DNA. See this blue patch. And in other homologous DNA there is a gray patch. Rest other is same. Okay, rest other is same. Just there is a patch in DNA. So this is how we can remember. This is a vertical product which is giving rise to patch. Means what I mean to say. See, this is a DNA. Okay. And here this is a A. Another is B. Another is B here. Okay. Then here will be blue. So this is a patch. Okay, so this is a patch model due to vertical segregation. Like this we can remember. Patch means non-crossover product, and there is a patch like thing we can see in the DNA. Patch of other strand, right? This is how the holiday junction is resolved. Resolve basically cleavage. Okay, resolvation of holiday junction means cleavage. Means this is the holiday junction, we want to cleave it. Either we will cleave in this direction horizontally or vertically. That's it. Now these are the proteins, or we can say enzymes, that are acting for this homologous recombination. So first of all, pairing homologous DNA and strand invasion in E. coli, it is Rec A protein, and in eukaryotes it is RAD51, RAD51 and DMC1 in meiosis. We have to remember what takes place in meiosis as well, which proteins are acting on meiosis. Now introduction of double strand break. In E. coli no protein, we don't know proteins. In E. coli this is used as a repair pathway only, we can say. But in eukaryotic protein, in meiosis, introduction of double strand break is done by this protein, SPO11. Okay, then processing DNA breaks to generate single strand for invasion, right? Tails, single strand overhangs, basically. This is formed by the Reg BCD helicase or nuclease. And the eukaryotic protein, MRX protein. Then assembly of strand exchange. Strand exchange is done by Reg BCD or Reg4, FOR4. And in eukaryotes, RAD52 and RAD59. Then holiday junction recognition and branch migration. Branch migration is done by RUAB complex, not well characterized in eukaryotes. And resolution of holiday junction done by RU roof C, RAD51, XRCC3 complex. So these are the types of protein. So basically we can remember like this. In prokaryotes, Reg BCD, then Reg A and RUVABC. These proteins are important. And in eukaryotes, Reg BCD homolog is MRX protein. Okay, Reg K, RAD51, and RUVABC is the not well characterized. Okay, we will see this with the help of diagrams. Basically, these important proteins are there: Reg K homolog RAD51, Reg BCD homolog MRX, and no homolog for RUAB. Okay, C is there. Ligase is there. Basically, XRCC3 is a ligase. Then RAD51C. Okay, and one extra protein, that is SPO11. This is for introduction of double strand breaks. Very important protein. Now let's see first in the eukaryote what take place. Now reg BCD. So we are considering this end as a double strand end. Okay, reg BCD enters a double strand break. Reg BCD enters. What is reg BCD? Helicase nucleus, right? Helicase nucleus. Where there is double strand break, reg BCD will come, right? So do note reg B, okay, reg B, reg D. These are the three separate proteins working in the complex. Okay. Reg B is a helicase, right? Helicase. 3' to 5' helicase, as well as nuclease. Okay. Then reg D is a 5' to 3' helicase. So both are this helicase, and reg C is doing a recognition of a chi site. So this is very important. Chi site. This is a chi site. See, chi sites crossover hotspot instigation. Okay, means this increases the frequency of recombination basically. Okay, so reg D moves fast and reg B is slow, right? Reg B is slow. Now as I said, reg B is a nuclease. Okay, so it is moving and cutting the DNA. See here, the DNA has been cut. Okay, so this is due to nuclease activity. Alright. So what is the function of helicase? As we have seen that this is unwinding the DNA. Right? See, DNA is getting unwounded. Loop growth. Okay, and here loop maximum. So what happens? This machinery means these three proteins are moving in the respective direction. As the tie site comes, speed gets low of reg B. See, reg B no load. Reg C we can see here. Then what happens? Formation of loop-like structure take place. Okay, see, loop-like structure is in place. Till the chi site, the nuclease activity is active. Okay, because what? The chi site will be recognized by the other next protein. Okay, so this chi site has to come out. Alright, so ultimate goal is to bring this chi site outside like this. Okay, so this is a formation of loop-like structure. As the chi site comes out, pause takes place. Pause means stop. Okay, again, now loop shortening. We can see here. Now this is also slower and this is also slow. At the end, we can see the chi site is, and there is a loop formation. Right? So we want to remember this function basically. Reg BCD is a helicase nuclease. Okay, reg D moves fast, reg B low, and reg C will recognize the chi site. Then the next protein will come, reg A. So reg A can be said as a homologue of the single strand binding protein. Okay, see now how this binds basically. 5' to 3' direction is fast. Active filament formation to coat 3' end of ssDNA. Okay, so this active formation takes place in the 5' to 3' direction only. Very important point. And other side, no filament formation take place. 3' to 5'. No filament formation takes place. So why this array K protein is there? Basically for strand invasion, right? Array K is a strand invasion protein. It also helps in pairing of homologous DNA, homologous chromosomes, as well as strand invasion. Then the next proteins, Ru A, B, C. Okay, first of all, A, B. These are for the, what we can say, Ru A, B. So A is for the recognition of holiday junction. Holiday junction, HJ, I will write here. And B protein is for the branch migration. Okay, so in the previous step, we saw that there will be formation of holiday junction. Okay, because strand invasion take place. Strand invasion means what? As we saw in the diagram, this is a strand coming down and it will move. Right? So this is done by the array K protein. Like this. Array K proteins will do this function. Same here. The other strand will be going upwards. Right? So this is known as the holiday junction formation as took place. What is this holiday junction? So the next protein, that is Ru AB, will come here. Ru A is recognizing the holiday junction. So these Ru proteins get attached at the holiday junction accurately as a tetramer. Okay, 4 molecules of Ru A. How 4 molecules? So we can see here. This is first, this is second, this is third, and this is fourth. So 4 molecules. And Ru B is 2 molecules. Dimer. Okay, dimer. This is 1 and this is 2. Alright. After this, Ru C will come. Ru C. Ru C, just 2 molecules, 2 proteins, right? Dimer. This is first and this is second. What it is doing? Cutting the holiday junction. Okay. Ru C can be said as resolvase. Resolvase. Okay. So Ru C, what does? Resolution of holiday junction. Got it? This is having a endonuclease activity. Endonuclease activity. Cutting the DNA, na? Cutting means endonuclease. Nuclease activity. Basically, this is an endonuclease. And Ru AB. So this B is a helicase. B is a helicase. Helicase uses ATP. Right? So this is about prokaryotic homologous recombination. Now let's see in eukaryotes. First of all, there is a introduction of double strand break. Very important step in the meiosis basically. So this is introduction of double strand break in eukaryotes. So who does this? Protein SPO11. Okay, SPO11. So SPO11 has a tyrosine. Okay, tyrosine. So this is a OH molecule of tyrosine which attacks the DNA, right? Two molecules of SPO11 are needed. So this is the first molecule, this is the second. Okay, and here we can see exactly what is taking place. See. So thus double strand formation takes place. Right? 3' OH gets free here and phosphate attached to tyrosine. Right? So this is our introduction of double strand break. Now let's see. Same process, nothing different here. First of all, double strand break by the SPO11. SPO11, see, we can see double strand break. As well as this is the other protein, MRX. Okay, MRX. Now, MRX, what is this MRX? Basically, MRX is the processing enzyme. Right, like reg BCD, giving rise to single strand tails. Okay, so what is this MRX? So this M stands for MR11, R stands for RAD50, and X stands for XRS2. So this is a complex of 3 proteins, giving the rise to the name MRX. Okay, MRX. Mr. X, we can say. So M stands for MR11, R is for RAD50, and X is for XRS2. So Mr. X, we can remember like this in eukaryotes, performing the work of processing, giving rise to the single strand tails. See. Now this will go inside, strand invasion. After 3' ends are formed, MRX is released out. Then the next protein will come, DMC1, RAD51. These are the strand exchanging proteins. Okay, homolog of Rec A, right? Rec A. See, filament formation is taking place. Assembly of the strand exchange protein filament. So strand exchange, or we can say strand invasion, taking place. So that's it. This is how recombination takes place in eukaryotes, right? Only 3 to 4 proteins are important. SPO11, which is introducing double strand break. Then is the MRX, Mr. X, right? Then is the DMC1, RAD51. Just 4 proteins. And in the prokaryotes, Reg BCD, Reg A, and Ru ABC. Right? So that's it for this video of homologous recombination. I hope this video helps you. If you find this video helpful, do let us know in the comment section.